There have been some very exciting changes. One is that medical oncology has matured and has become one of the established specialties of medicine, and that we have coordinated better the interaction between the various specialties. And I think we have made great headway in incorporating preclinical progress, preclinical research and translational research into one single direction in which we are going. And I think that was not easy. We think about it today as, well, it's done. It's not quite done, but it has been done quite substantially. And I think it has reflected well on the care of our patients, and I think it has accelerated research because we are able to take observations back to the lab and from the lab to the clinic.
Progressing to endocrine-based therapy. So, do you really think that we need targets to improve or to treat our patients today? Maybe in the future, but today?
Yes and no, because some progress are here, like our ability to identify what type of tumors of patients should receive hormonal treatment or chemotherapy. And for instance, the genomic signature emergence are here. And those type of signature, it is not exactly a pure 100% target, but it's a better definition, identification of what type of treatment we should give to our patients. And those signatures emerge everywhere for chemotherapy, for hormonal treatment, for the need of radiation in situ carcinoma, for instance, in colon cancer also. So some progress here. It's the relationship between our targeted treatment or our treatments and a clear tumoral target is not here in every case. Right. But it's probably the way to go, and we are right now in 2013 at the beginning of this new step in the treatment of tumors.
You are still remembering the bad news. I mean, I think that we are on the way. We have found very spectacular targets in breast cancer. I think that HER2 is the clear example. And we have still a very large room for new targets. And what we need is to identify these targets, and mostly to define or design the good drugs that could block these targets. But finally, what we can call HER2 negative population becoming endocrine resistant, there's a large population in breast cancer for which it's clear we don't have a good target.
Exactly.
But it should work now. I mean, this is one of the next task for the next 10 years. Clearly, it's not the problem in the HER2 domain. We will continue to improve drugs, but in a very clear idea, we have done a lot of efforts, and we have reached extraordinary objectives in the HER2 positive. The problem now is that for HER2 negative, we failed to observe any strong molecular predictor out of estrogen receptor.
So we have been much more effective at developing therapeutics than developing diagnostics. And I think where we really need to concentrate and focus in the future is in identifying biomarkers. Because without biomarkers that actually enrich a population, we don't really have targeted therapy, right? Right. So we have identified a number of potential targets. Antonio is absolutely correct, but we need to do a better job, even in the HER2 positive population, because HER2 is not a very good predictor. Its absence is an excellent predictor of no benefit, but as a positive predictor is weak. So emphasis on biomarkers and the work of individuals like Joan is critically important. We think very simplistically of targets, molecular targets. How about the importance of the target in relation to the context? So for instance, is a PI3 kinase mutation the same in ER-positive breast cancer or HER2-positive breast cancer or in stomach cancer or in some other tumor?
I think this is a very good point because right now we're just testing a target and not seeing the context, and maybe we know very well that every target may be good or bad depending on the cell context. So I think that one thing that we have to evolve in the concept of personalized medicine is not just test the target, but test the pathway, if this leads to pathway activation, not only if the target is present or not. And I think this is where the new technologies can help to really identify activated pathways and then select the critical player in this pathway. But this can depend in different tumor types and within the same tumor type. So I think that the main challenge ahead is what we call individualized medicine. Today we are doing stratified medicine at best. Mm-hmm. But I think that the better knowledge on pathways, the new technologies, maybe in the future will lead to this more personalized medicine, not based on only the target, but pathway and context.
We have seen in the last year many phase 3 clinical trials which have been clearly negative. And in my opinion, some of them have been just because of the clinical trial design. I think that if the industry would work together with us, we might improve or we might, I don't know how to say, we might maybe help them to design better clinical trials. Do you think that maybe, Joan, Tony, maybe the academic organization might help companies, or might help cooperative groups to design better clinical trial design, to improve the clinical trial designs, and to have better drugs in the market?
We should help as academia to the pharmaceutical industry to develop, at least to put in place, a structure of very quick trials in which we could answer very quickly, probably not clinical answers, but biological ones. I think that there are still very few open window opportunity trials, and even very few neoadjuvant trials, but probably the very short trials in breast cancer, the window opportunity as something that could help in the future to the pharmaceutical industry to take much quicker decision with a better drug.
It is not just clinicians, it is clinical trialists, because there is not only an art but a science in developing the best clinical trials. Along the points that both of you have made, I think we are also facing another challenge, and that is that if multiple companies have drugs of the same family, it is not necessarily the best drug that will get ahead, but it's sometimes the drug of the company that is more aggressive or bigger or richer. And we have had several recent examples of how, because of that phenomenon, the phase 3 trial fails, for instance in Iparib, although there are other examples. And I think something that the pharmaceutical industry has been somewhat resistant to is for the academic community to help them select before getting to phase 2 or phase 3 trials. You're correct, there are, I don't know, several dozen PI3 kinase inhibitors or inhibitors of that pathway. Nobody except the company that develops them gets access to them in a meaningful way to compare them to all of the others. And we often end up killing potentially good drugs and testing drugs that perhaps do not deserve to be tested.